y'all, here we are back again for another exciting edition of Misconnect and Biology. You know, I'm thinking maybe I should contact the people at Netflix. They might, want to buy a little piece of this. I could be like Bill Nye the Science Guy, but I could be like Miss G the Science Chick. I don't know. I think I'm onto something here. You guys come up with a good theme for me. All right, so today we're going to talk about ways to move things in and out of a cell, because a cell needs to be able to get nutrients in, and it's got to be able to get waste products out, because we don't want to keep all the waste products inside, and we definitely need to get nutrients in so that the cell can feed itself. So when you're thinking about transporting things in or out of a cell, there's two main things you need to think about. The first one that you need to think about is the particle size. There is a big difference between something like water and making sure, that water can get in and out of a cell, and something like glucose. Okay, so if we want to move something into a cell, you could move this in, or you could move this in. So it's kind of the equivalent between moving, like a lamp into the house, and then moving a king-size bed and dresser all together into the house. Getting things in is going to be a little bit different. So how big is the particle we're talking about? Carbon dioxide, also pretty little. Diatomic oxygen, also pretty little. So we have some smaller molecules, but then you're also going to have some bigger molecules that have to get in and out. The other thing you're going to worry about is concentration. So how much is there of that substance outside the cell compared to inside the cell? Or how much of that substance is there inside the cell compared to outside the cell? So what's the concentration difference is going to be really, really important to you. Permeability is another idea. So cell membranes are what we call semi-permeable. So semi-permeable membranes, that means that some things can get in it and some things can't. So permeability just refers to anything being able to get in and out. So a completely permeable membrane would mean anything can get in and out any way it wants to. Cell membranes are semi-permeable. Some things can get in, some things can't. So we've got a membrane here, and I have some little orange particles in there, and those little orange particles will fit through the holes. They can move through the spaces in the membrane. They have no problem. They'll go in and out. But these green particles are bigger and fatter. They can't get in and out of the membrane. So the cell membrane is impermeable to the green molecules. Now I have a cool little model here to show you the same idea. So here we are, we have two different particle sizes in here on the membrane, and we have a high concentration on this side of the membrane, and we have a low concentration on this side of the membrane, and that's for both of them. So we have a high concentration of the red molecules here, and a high concentration of the white molecules here. We have a low concentration of both the red and the white molecules here. Now, this membrane, you can see, is semi-permeable. It might just look permeable right now, but when I turn this and give it a little shake, you can see that this membrane is permeable to these white molecules, but it's impermeable to these red molecules. So now I've changed my concentration difference. So I have a high concentration of white molecules over here, a low concentration of white molecules here. There's still some white molecules, but it's a low concentration. And then I have a high concentration of red molecules here, but no concentration whatsoever of red molecules over here, because they couldn't get through the membrane. So again, this membrane is semi-permeable because some things can make it through the membrane, but other things can't make it through the membrane. So, words to describe solutions. We've talked about solutions. You know what that means. That means that we've got a solute and a solvent, and we have the solute can be anything. It can be salt, it could be sugar, it could be glucose, it could be oxygen, it could be carbon dioxide, it could be anything we're talking about. The solvent is almost always water when we're talking about living things. So for the most part, we're talking about water. So I have this happy little cell who's going to help us through these problems, and we have this happy little cell, and he is isotonic with his environment. So there's tiny little polka dots in that diagram, and that is representing what those are all representing little water molecules, okay? So we're saying the concentration of water outside the cell is the same as the concentration of water inside the cell. So as long as those two levels are even, then we say that this state, these two solutions are isotonic. So the cell is in an isotonic environment, meaning the concentration of water inside and the concentration of water outside are the same. The concentration of solute and the concentration of solute, the concentration of solute inside, concentration of solute outside are the same. Okay, so now we've thrown some solute in. We had water and water, now we've thrown in some solute. Okay, so let's call those hydrogen atoms. The hydrogen atoms are on the inside, hydrogen atoms are on the outside. They're in equal concentrations. So this cell is at a state of equilibrium. So the solutions are isotonic, but the cell is in a state of equilibrium. Okay? That's an important difference. So put a little star next to it so that you remember to mark the difference between isotonic and equilibrium. Because isotonic is an adjective that describes the two solutions, and equilibrium is a state of being for the cell to be in. All right, now, oftentimes we are not at equilibrium. In fact, if your cells were at equilibrium, that would mean you were dead. So it's better not to be at equilibrium. But there are situations that are the opposite, which is good when we're alive. And so we have the words hypertonic and hypotonic. So hyper means above or more, like somebody who's hyperactive is overly active. And so hypertonic means we have a higher concentration of something. So hyper means high. So hypertonic means where we have a higher concentration of solute. Okay, so unless we are specifically describing water, we have a high concentration of solute. Okay, so if we look at our diagram the way it's drawn right now, inside the cell the solution is hypertonic, and outside the cell the solution is hypotonic. Now hypertonic and hypotonic, hypo means below, hypertonic and hypotonic are opposites of each other, but they're also relative to each other. So it's like saying taller and shorter. So I can't just say I'm taller. Okay, because I'm taller than who? I'm taller than Eloise? Yes. I'm taller than Mr. Burnside? No. Okay, so you always have to be talking about what something is hypertonic to. So the inside of the cell is a hypertonic solution. The outside of the cell is a hypotonic solution, and they are in comparison to each other. So if one is hypertonic, you have to say what it's hypertonic to. Okay, so hypotonic outside the cell, hypertonic inside the cell. Okay, so it's not always that way though. Sometimes outside the cell can be a hypertonic solution and inside the cell can be a hypotonic solution. So you can't always memorize, oh, inside is this, outside is this. You have to look at the situation because the situation can change based on the environment the cell is in. So wherever you have a higher concentration of solute, that's where you've got a hypertonic solution. Wherever you have a lower concentration of solute, that's where you have a hypotonic concentration. So here we go. Right here, the top part of this cell, for the red molecules, hypertonic, hypotonic. But if we're talking about the white molecules, hypertonic, hypotonic. So even as we're looking at individual molecules, it may change. So you have to know which solute you're talking about too. All right, diffusion. So diffusion is passive transport. And what that means is it does not require energy. No energy is necessary for diffusion to happen. It just happens. It's going to happen spontaneously, and it's going to move molecules from a high concentration to a low concentration. So this thing that I keep showing you, this is diffusion. Molecules moving from a high concentration where there are lots of them to a lower concentration where there are fewer of them. So again, here's a low concentration, here's a high concentration, and by diffusion, they're going to move. Now, I'm applying some energy, but that's just because of the nature of the model, okay? But they will naturally just move through from high concentration to low concentration. The ultimate goal of diffusion is to reach equilibrium, to reach that point where you actually have equal concentrations on both sides of the membrane. So we're not reaching, actually, now we're at equilibrium. And so that's our ultimate goal, is to make it to equilibrium where we have equal concentrations on both sides of the cell. Remember that when we are at equilibrium, our two solutions are isotonic, which means compared to each other, whatever our solute is, those concentrations are equal. Okay, so if our cell looks like this, the inside of the cell is hypertonic, the outside of the cell is hypotonic. Those hydrogen atoms want to get out of the cell. Luckily, the cell membrane is permeable to those hydrogen atoms, and so they're going to move out, okay? But when we have this other situation, sometimes the outside is hypertonic, the inside is hypotonic, and so those membranes are going to want to move in. So anytime you're those molecules are going to want to move in. So anytime you're talking about the movement of the molecules, you have to tell me where they're moving from and where they're going to. So in this first situation, the hydrogen ions are going to move from inside the cell to outside the cell. And in this situation, the hydrogen atoms are going to want to move from outside the cell to inside the cell. You can't just say, oh, they'll move inside. I don't know what that means. Oh, they'll move outside. I don't know what that means. From outside to inside or from inside to outside? Which way are they going to move? All right, there's also something called facilitated diffusion. And facilitated diffusion is still passive transport. That means it still doesn't require any energy. It still happens spontaneously. It still moves molecules from high concentration to low concentration. The ultimate goal is still equilibrium. The difference is that it requires protein channels. So you have to have a special way for those molecules to get from one side to the other side of the membrane. And so there are these special protein channels that are created there, and just the molecules can move right through. They're not going to slip through the membrane, the spaces in the membrane. They have to go through these protein channels. The protein channels are specific to the molecules that they help move in and out of the cell. So one particular molecule can only go through a particular kind of protein channel. And a particular kind of protein channel will only allow one particular kind of molecule to go through it. Then there's osmosis. Osmosis is cool. You've probably heard of Osmosis Jones. Yeah, I don't know why his name's Osmosis Jones. As far as I remember, he has nothing to do with osmosis. It's just a science word. Okay, osmosis, still passive transport. Doesn't require energy. Happens spontaneously. Osmosis moves water molecules from high concentration to low concentration. So osmosis is the movement of water. It's specific to water. Water molecules. Osmosis, water. Osmosis, water. Osmosis, water. So all we're doing is we're giving water its own name for its facilitated diffusion. Okay? The ultimate goal is equilibrium. It's just a specific type of diffusion, and it is facilitated diffusion that refers to the movement of water. Now, osmosis requires protein channels, and they are protein channels that are called, wait for it, biology people worked really hard on this, aquaporin. They are pores that move water, so they're called aquaporin. So aquaporin is a protein that helps move water molecules across the membrane. So we said with facilitated diffusion that protein channels are specific to the molecules they move. So aquaporin moves those water molecules. So osmosis sometimes solves problems that we can't solve on our own, that the cell can't solve on its own. So let's take this example. So we have our little dude here, okay? He's got a lot going on. He's got all these blue particles inside of him, but those blue particles can't get through the membrane. So ordinarily what would happen is plain old regular diffusion would just move some of those blue particles out, and we would end up with even things at even concentrations on both sides of the membrane. But they can't because they're too big. So what's going to happen then is the water is going to solve the problem. Water is going to move to the inside of the cell because you can dilute out that solution by putting more water in it. So think about having like really strong Kool-Aid. You mix up your Kool-Aid and you're like, oh, I put in 3 cups of sugar instead of 1 cup of sugar. What do I do? How do I make this not so strong? Well, you dump more water in it. Okay, and you dump more water in it, it gets more diluted, you're good to go. Okay, the problem though is that there is a cell membrane there. And so you're gonna move that water into the cell, but then the cell is gonna get bigger and bigger and bigger and bigger and bigger, and it's gonna push on that membrane. And ultimately, don't tell the cell this, it could explode. We don't want that to happen. Okay, sometimes though we have the opposite. Sometimes we have a hypertonic solution outside the cell and a hypotonic solution inside the cell. Okay, so what's going to happen? Make your prediction. What do you think? Too much solute outside, not enough solute inside. Yep, water is going to move from inside the cell to outside the cell so that it can dilute out that concentration of solute outside the cell, and our cell is going to shrink up and get very, very small. okay? So you're gonna end up with a shrunk up little shriveled cell. That's so sad. All right, then we have active transport. I promise we're getting to the end here. Okay, well, in a little bit. Active transport is not passive. That's why we call it active transport. Active transport requires energy. Active transport happens according to the cell's needs. So active transport isn't going to happen because there's a concentration gradient, because there's a difference in concentrations. Okay, so remember passive transport, we had a concentration gradient. We had high concentration to low concentration, and those molecules moved down the gradient. So we went from high to low, and they moved down. Okay, that's not what's going to happen. We're just going to go according to whatever the cell needs. Again, active transport not related to concentration. There are a couple different kinds of active transport. There's molecular active transport. There's something called exocytosis. And there's something called endocytosis. And endocytosis comes in two kinds too. Endocytosis has phagocytosis and pinocytosis. Okay, these are all words you should be able to say. Molecular active transport, endocytosis, exocytosis, phagocytosis, pinocytosis. Molecular active transport. Okay, so molecular active transport is going to move molecules using active transport, so we're using energy, and it's going to use pumps made of proteins to move the molecules across the membrane. Now, it's not like a protein channel. A protein channel is really just like a straw, and then the molecule can go straight through. Okay, so we've got our protein channels for facilitated diffusion, but molecular active transport uses a pump. And what happens is that the molecule is going to attach to the protein pump. The protein is going to change shape. It's going to recognize it and go, oh, hey there, hi there, ho there. Okay, and the molecule is going to get let in, and the shape is going to change again, and then the molecule will be able to attach. So look, here's my tape molecule. The tape molecule docks, and the protein goes, oh, hey, how you doing? And lets the tape in, and then changes shape again, and then the tape can go into the cell, okay? And then later on, it'll go back to the cell, more tape will dock there, okay? It'll allow the tape in, and then drop the tape out. That was a super high-tech demonstration. I hope you're impressed. Remember to tell Netflix about that. There's also something called exocytosis. Exocytosis, exo means out. Cyto means cell, so exocytosis is out of the cell. And so with exocytosis, you're moving things out of the cell. So what happens is that the membrane of a transport vesicle just merges with the cell membrane because they're both made out of the same thing. They're made out of that phospholipid bilayer. They're made of exactly the same thing. And so all that happens is this little membrane right here of this vesicle moves over, it bumps up, it's like, hey, scooch over, make a little room. And so the cell membrane makes a little room, and this merges here, and we have exocytosis, and then it's basically just spews its guts outside of the cell, and that's how you can spew lots of different molecules out. So you can either do this for a really, really big molecule, a molecule that's so big that it can't get out of the cell on its own, or you can do this for lots and lots and lots of molecules. So if you have a whole huge mess of molecules that need to get out, and it just wouldn't be efficient to move them out through protein pumps, or maybe they're charged, maybe we're talking about charged things that you can't get out, exocytosis is going to do it for you. And then there's endocytosis. Endocytosis is just the opposite of exocytosis. So exocytosis takes things out, endocytosis brings things in. Endo means in, like endoplasmic, inside the cytoplasm. Endocytosis, bringing things inside the cell, okay? So again, we have our molecules that are gonna push up against the cell. So here's outside the cell, here's inside the cell. Our molecules push up outside the cell. They keep pushing, push, 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 okay? And so you see that the cell membrane starts to fold in, and then they push, 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 push. And as they push in, these two parts of the membrane start to come together, and then you get this transport vesicle of its own. Again, that's why a transport vesicle is made of the same membrane as the cell membrane. All right, here we go. Endocytosis, two types. Super easy though. Phagocytosis and pinocytosis. The only difference between them is what it is that we're bringing into the cell. Phago means to eat, so phagocytosis means that cells are bringing in solids. So phagocytosis is cell eating, solids being brought into the cell. Pinocytosis then, pino means to drink. So pinocytosis is when the cells bring in liquids. So phagocytosis, solids, pinocytosis, liquids. That's it. What are we bringing into the cell? So we're identifying what it is that's being brought into the cell. All right, let's watch some videos.
Small, non-charged particles, primarily gases such as oxygen and carbon dioxide, can diffuse through the plasma membrane by moving in between the phospholipids in the bilayer. However, the cell needs to control what enters and leaves, and so transport proteins aid in the selective movement of other molecules across the membrane without the input of energy. Through facilitated diffusion, larger molecules, polar molecules, and charged ions use channel proteins embedded in the bilayer. The transport of other substances requires a special carrier that will bind the substance on one side of the membrane, which triggers a conformation change in the protein carrier, causing it to release the substance on the other side. When the substance being moved across the membrane is water, the process is called osmosis. The cytoplasm of the cell, as well as the interstitial fluid, is composed of solutions. The solvent, usually water, moves across a semi-permeable membrane toward a higher solute concentration, consisting of various molecules or ions, until equilibrium of the solutions is reached. The plasma membrane contains proteins called aquaporins, which are specialized channels for the movement of water during osmosis. A cell in a hypertonic environment will have water move from the inside of the cell toward the higher concentration of solutes in the solution outside the cell. In a hypotonic solution, the concentration of solutes is higher inside the cell than the outside environment, so water will diffuse into the cell. When the solutions on either side of the membrane reach equilibrium, they are referred to as isotonic. When substances need to be driven against their concentration gradient from low to high concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier, common in most animal cells, that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside.
Okay, guys, that's it. I got nothing else for you. Work on this because it's hard.